Okay, thank you, panel members and audience and my collaborators. It's really my pleasure to update on our project on the functional bone regeneration in challenger bone disorders. Yeah, so this is the outline of the study. Basically, just to refresh our memory on what we are going to do. And then so far what we have achieved, and then future studies and plan of the project. Now, population is aging. It's really a challenge issue, particularly for our musculoskeletal systems. This affects Hong Kong much more because we have the longest lifespan for male and females. So clinically, we see more and more challenge cases in old people. Including the age-associated osteoporosis, its clinical complications, its fracture. So its repair is much more challenging compared with our younger patients. Currently, our methods to treat the younger patients is satisfactory, while it is really challenging for old people, especially with delay and eventually non-healing of those fractures. And avascular necrosis is also a big challenge. We can still recall the SARS years. We used corticosteroids, not only for those SARS patients. Now, COVID-19 may not so severe compared with SARS, but we have a lot of other indications. And then in sports medicine, either for young or older people, we have ACL injury, which is really challenging because it's healing between soft tissue and hard tissue, and we see the tunnel widening that affects the treatment outcome. So we also understand, based on our summary in a few monographs edited by myself and my colleagues, basically for the healing, for all these three conditions, it's affected by many conditions, including age, for example, and metabolic conditions, and if people have disease or taking medications, that also affect study outcomes. So optimally, we can have a really restoration of original structure, but if somehow, due to certain reasons, we have scar tissue formation, and that affects the healing quality and the restoration of our daily life. So we have some common features here in osteoporotic fracture, AVN, and ACL injury, including the quality of bone decreasing, and bone marrow stem cells quality and the quantity also decreasing. Now the MSC differentiation, we see the osteogenic and angiogenic, those are coupling events during restoration also down, and adipogenetic potential is increased. All these are negative. So they share the common feature. And if we look into the healing progress, basically the undergoing cascade reactions from inflammation, repair and restoration and remodeling towards the normal for all these conditions. So MSC is very crucial, the bone marrow stem cells. So how to control them towards the osteogenesis and angiogenesis are crucial for us. And now in our orthopedic surgery practice, we know not only the surgery itself is fundamental, but it's biological enhancement and rehabilitation, including the use of biophysical enhancement during the immobilization phase, which is really essential. All these factors that can generate really functional restoration of musculoskeletal tissues. So for simple illustration, this we see we have a bone defect. We need biomaterials for bone the defect replacement, and then we sometimes need incorporation of cells and growth factors. And after surgery, as mentioned, we probably need a certain physical intervention to give mechanical stimulation. Now the mechanobiology is very hot in this area. We realize such their importance. So in our previous efforts, we did one aspect for each project, including identification of the growth factors, and then those from either the endogenous growth factors or exogenous, just like from herb medicine, from mineral. And we also developing biodegradable materials. Professor Yu Jian is our core PI. It's famous in this area. So now tissue engineering come to our attention, basically this incorporating the structure and the cells, the growth factors for the enhancement of the fracture repair. So in our project, we incorporate all these three aspects for those challenging musculoskeletal repair for avoiding, as I mentioned, for those challenging conditions with potential delay and eventually non-healing that generate a lot of trouble for old people, also our healthcare burden. Now, for this project, we have three phases. Basically, first of all, to optimize the implant design for our applications. Second is to conduct in vivo preclinical tests to validate our design using small and large animal models, which are required by regulatory bodies. And thirdly, of course, we try to get those safety approval so that we can prepare for our clinical trials, eventually get our innovative products to be registered for wide clinical applications. So this is our overall concept from cells up to matrix using moderation and growth factor supplementation biophysical interventions. Those are detailed designs. And for in vivo study, of course, we, as mentioned, using small and large animal models, we have to do grouping, sample size estimation, and sophisticated analysis using conventional cellular, molecular, histological, and biomechanical testing. Those are standard required by regulatory bodies. So the same, after using small animal models, we can identify which kind of combination could be optimal for us to go for large animal studies, which are quite expensive. So now, before going to clinical, multi-center clinical trials, we have to understand what are requirements required by regulatory bodies. Basically, we are following Chinese regulatory requirements, so-called National Medical Products Administration, used to be the CFDA, China FDA. Okay, so we have to follow all those procedures to satisfy the regulatory requirements, including those safety tests following, generally speaking, the ISO standards, international standards. So after all this, we hope that we could reach the stage that we get safety approval so that we can move towards later on for the clinical trial. We form, as I mentioned, this is really a multidisciplinary approach. So we have surgical enhancement, biological enhancement, biophysical enhancement as an integration. So I'm overall leading the groups, but we have experts from material sides, from biological aspects, and also from biological intervention from various local universities. And this is the job description or distribution among individual territory institutions based on their strengths. So following the advice from our panel members, basically we have our clinical advices, and we have from each areas, we have the leading investigators in this, and we also have international collaborators or advisors who may give us feedback and adjust our efforts so that we can move towards really regulatory body testing results, requirements. So at the very beginning, starting with that, we can gather together, and we have regular meetings every 3 months to update our progress, because we have separate, efforts, and we should coordinate well. So then, with the time, we have to move to the zone because of the COVID, right? So, but we still keep on. Really, those gathering, physical gathering is very important. Apart from currently the exchange of the information, we really conduct a few locally and also internationally that help us towards current achievements that I'm going to update with you. So according to the nature of the project, we have also the obligation to train the scientists. So quality control is very important in conduction of all those experiments. So we coordinate all that into the training programs. We have annual training events from analysis technology towards the animal experiments. We have to use the international standards to train. So this is the program we developed together among our investigators together. Really, we appreciate the grants that can unify our efforts locally towards a systemic approach in musculoskeletal research. Now, quite impressive at the moment that we are leading these areas by establishing certain guidelines that eventually the national standards, the NMPA, also consider our recommendations. So this is a general distribution according to the stage and the years. Okay. So we conducted some site visit before our public forum today. So basically, we have certain deliverable. We committed for publications, conference to be organized, and so on and so forth. And we conducted a second visit 2 years ago. And then we hope that after this stage 5, we are going to be able to finish the whole project in time. But because of the COVID, we did have the challenge. So up to now. Concerning the achievements, basically we committed at the very beginning in the proposal, for example, concerning the number of publication 10 to 5. For the first, the second, and the third visit, so far, we really far exceeding the requirement, so commitments so far. But that is also good for us to train the students that they need get scientific training and also recognition in terms of publications. And the scientists, so far we trained approximately up to 40 for local and regional people. Those are just a snapshot of representative publications generated so far from our project. Okay. Innovation is important. We need to protect it. So far, we already obtained, for example, invention patent and US patents, and also a couple of them are still under, evaluation at the moment. So we have so far trained from CHK, CTU, PolyU, and Hong Kong here. Then those are the young scientist students graduated. Some of them are still with us, and some of them already, have their new challenge, making their new career. So for R&D, we also participate actively into the local competition and also form some spin-off to carry on our collective efforts. Some have been internationally recognized, for example, in the Geneva Invention Awards, and reported locally on our collective efforts. So at the moment, we have achieved from all aspects with representative publications. One from material scientists. We generated quite controllable biodegradable materials by Professor Liu's group. and published also recently on their new technology and also their inventions so that we can make our innovative device. And this is his paper on nano-dual phase metallic glass film enhanced strength and durability of nano-granite magnesium alloy, which really help us to solve our challenge clinically to make implant more durable and stable. And for biological parts, we are using small animal models, and one of that is also carried here in Hong Kong U by Professor William Lu. And we trained also the students who are from traumatology and sports medicine by establishing small animal models, as mentioned, by developing scaffold materials, identify those bioactive molecules, safe ones, for application using the model ACL reconstruction model. Now, at the very beginning, we try to identify those bioactive growth factors. So we have to do a lot of screening by combining various growth factors with the enhancement of the MSCs towards osteogenesis and angiogenesis lineage. So we have already published the relevant data and then identified those for carrying towards the integration into biomaterials that has been developed for the bone defect repair. So those are 3D printed scaffolds mimicking our trabecular bone in our body, where we sustain more osteoporotic fractures. So then we also develop HM hydrogel. Those are really relevant for us to carry in those growth factors to make them active after implantation. So we developed AVM models by injection those corticosteroids mimicking clinical scenarios, and then we implanted those materials into the drill hole to observe their bone regeneration. Indeed, after all these serious experiments, we identified that those growth factors we pre-screened at the very beginning, after incorporation, that finally we achieved the best osteogenetic potential. So by looking at the regenerated tissues. And then we also used ecardine that purified from traditional Chinese medicine, currently used for treatment of osteoporosis. We identified that active compound after incorporation into our scaffold, and indeed we see that after incorporation, the ecardine and the secretome from the stem cells, we achieved the best effects as well. So we carry further. After implantation, we used ultrasound to simulate mechanical stimulation for those tissues, because bone musculoskeletal regeneration is slightly different to other organs. All those systems are under permanent mechanical stimulation. We have to simulate that. And indeed, we find that under the influence of pulsed ultrasound, we can maintain the properties of the implanted bioactive materials and make them even more osteogenic and angiogenic for achieving better repair. So these are those small animal experimental models simulate clinical conditions. Now, we further look at the challenge part. So commercially available ultrasound can only treat the superior tissues, not very deep, but osteoporotic fracture like a hip are deep bones. So with help of our scientists, engineers, Professor Zheng Yongping, for example, ultrasound experts here, we are able to develop those unique ultrasound devices to satisfy our purpose to treat deep bone fracture repair. So then for biomaterials, biodegradable magnesium and alloys, we have developed magnesium wires. Okay, it's durable wires for fixation and suturing the tendon into the bone kernel, which then after implantation it can degrade, and in fact those bone kernel can be maintained. As I indicated in the very beginning, clinically we have a big challenge after implantation, insertion of the tendon into the bone may be resorbed, the tendon can be widened, so that mechanical stability is affected. Now with that, we can have really very tight conjunction between the tendon, soft tissue, and the bone. So this one revised and pending for acceptance. I'm very pleased that the day before yesterday, we got the paper accepted by the Journal of Magnesium and its alloy. So we demonstrated that the interface is like the fibrocartilage, the normal structure restored, instead of the gap in between, suggesting that the healing really being enhanced with restoration of the interface materials. So another challenge part in osteoporosis is that many people now taking bisphosphonates, the drug, anti-osteoporosis. Very good, they can prevent the osteoporotic fracture, but somehow there are some portion of those patients still suffer from fractures. Not in the conventional side, distal radius spine, but in the proximal femur, in this region. This region is very difficult to heal, and particularly under the influence of bisphosphonates. And we identify that the mechanism behind is that the bisphosphonates can induce fibrosis in between during the healing that prevent the bone normal healing and remodeling. So by using the magnesium, we identified its mechanism is using the single cell technology. We identify magnesium ions is very effective to prevent the fibrosis formation. And then we also test in other models. Basically, in musculoskeletal and also others tissues healing, fibrosis is bad. So if the material can have the function anti-fibrosis formation, it can really have great potential, including the SCI, spine cord injury, the nerve repair. Because of fibrosis, they cannot normal repair clearly. So we identified the novel mechanism and then just published this year in Materials Today, and then as research highlights in Science. So we are really inspired by our findings and carrying forwards from small animal, large animal, and we are confident that now moving towards the clinical tests later on. Okay, so eventually we move towards big challenge of the bone defect, big gap. After incorporating the bioscaffold materials made of magnesium in cooperation with Professor Liu, and we see here, if no insertion of the biodegradable magnesium, the gap remains. But insertion of the magnesium, you see this gap being breached by the normal tissue. This is really very inspiring and encouraging for us to develop cost-effective novel implants. And we also published all this data and also get mentioned the innovation inventions. So what is the underlying mechanism, the signaling pathways between cell and the matrix? Professor Yuan sitting also here, developed a lot of technology to identify, for example, the Wnt beta-catenin signaling pathway are very important in bone regeneration process and also published accordingly. So over the past few years, we did make efforts scientifically to confirm what are the underlying mechanisms for those our inventions for growth factors and the biomaterials, bioactive materials. Using experimental models, we explain a lot of phenomena, but still we no, there are still other mechanisms behind. For example, during degradation of magnesium, there are also alkaline environment being generated during degradation and hydrogen gas released from degradation process. If that is controlled, we know all these three factors, magnesium ion, the ions, the hydrogen gas, and the alkaline environment are favorable for osteogenesis. So this is a very unique materials now we are using for developing innovative medical devices. And there are also others we hypothesized and to be confirmed in coming research projects. Because all these are so important for us to design the desired products with biosafety and clinical safety in terms of achieving better treatment. So for large animal model, currently we are not able to do here in Hong Kong. In Shenzhen, we identify a big center for doing those animal models. So we carry on the goat model over there using ACL model in goats, and we identify those similar results, similar to the small animals. And for magnesium implants, you will see here control group using conventional intramedullary nail. The callus formation is minimum, but after incorporation, our magnesium into the intramedullary nail, you will see here the callus formation is very impressive. Later on, remote it to more normal, while here in the control, you still see the gap. So I think our innovation really satisfies our initial design based on our understanding on those advantage of materials. So for avascular necrosis, we tried quadrupedal animals, but they never developed to joint collapse. Now, we have moved to bipedal emu model, right? Also across the board in Shenzhen. And we developed those stand for fixation of the hip. And we identify one technology is very good for our applications. We published that as well. So using this model, we developed the stand and also the injectable cement. After injection of the magnesium-containing cement, we found that this is the overall concept, right? What we found that in normal condition, this is normal. This is a joint collapse. After using the magnesium stand and the cement, the incidence of collapse is significantly reduced compared with normal. So it's almost 80% reduced, which is also very encouraging. All this help us to build up the basis for carrying over for clinical tests by following the NMPA standards. So over the years, we got all this safety approval, and this is all the safety report from the NSF certified testing center in mainland, and all of those are results. So we so far got. And here locally, our surgeon got also the human ethics approval to be ready to carry on certain clinical studies locally. And of course, for the future, the challenge part, we have to understand the interactions further based on the materials we already got from large animal experiments. And then cell matrix interaction signaling process will be further processed by Professor Yuan's group. And the ultrasound device, we have already applied for the patents. And now, so far, we got over 100 publications. The challenge, as I mentioned, is that COVID, because that prevented our free travel to mainland institutions. But so far, we have a team based on there. So all this helped us to, avoid further delay. Now, we applied for extension of completion of a project for 8 months because of the COVID. Otherwise, we complete according to our original design. So very pleased that you approved our applications. We hope that we can provide further scientific data to be published as well and for final reports. So for the parts not belong to this scene-based, but I hope that I can carry on, is the challenge that the regulatory systems. Locally in Hong Kong, we do not have regulatory body to approve drugs and medical device. So we have to follow the mainland system so far. Hopefully, we can really work with the hospital authority to establish such regulatory bodies locally, but I know the challenge is very big. So I just proposed the one that we can develop together the integration of the system in at least the Great Bay areas. So with that, we can really go for later on the clinical trials. So this is not part of the scene-based, but I hope that I can still carry on further to work with manufacturing side, really to produce clinical devices for clinical applications. So far, our team are very happy about our collective efforts and achievements so far, and we hope that we can speed up all this to complete our project within the extension period of time. Okay, thank you for your attention and questions.
So thank you, Professor Ting. Any questions from the floor? Yes, please.
Thanks, Professor Chen, for a very nice summary of what great work that your team has accomplished so far. Looks like you've made a lot of progress with the magnesium aspect of it, and you also touched upon the biologics, which is another focus of your group. In terms of delivering the biologics in conjunction with the materials or the bone cement, and do you have any insights on what is a good way, the best way to deliver a growth factor? Is it, you can think of recombinant proteins or messenger RNA or gene therapy, and what is the best way to accomplish what you want to accomplish?
Okay, thank you. Concerning the later on the clinical applicable devices, we can only apply one defined treatment. For example, although functionally we try to have the cells, growth factors, materials, but regulatory body said no, we cannot approve that. You need to either do the biologics or biomaterials. Okay, so based on this research, we move more faster for the biomaterials. That is rather controllable. And concerning your question on the biologics here, the incorporation of the growth factor for application is really tricky. So we have two arms. One is the autologous and another, endogenous and exogenous. So for exogenous, we have either the mineral ions or the TCM herb active compounds. All those are temperature insensitive. So basically, we can use normal fabrication devices or just at a room temperature. Eventually, we have low temperature devices to fabricate together the growth factor incorporating the material by building up the 3D structure. So we proved that they can keep for 2 years without any loss of the function of those exogenous growth factors. But for BMP-related endogenous growth factors, really they are temperature sensitive and also agent sensitive for forming the materials. So we compared with the fabrication process, and finally we found that the two ways. We use the growth factor, endogenous growth factor proteins on the one side, material on the other side, and we immerse before implantation. Then we can achieve what we wanted, instead of we make one ready products. Thank you.
Thank you, Professor Ting. Any other questions? So if not, thank you, Professor
you.
Ting, for your exciting talks.